I'm delighted today to be given an opportunity to talk about new treatment strategies, what's in the pipeline for Parkinson's disease. And I think this is probably something we need to work out what we mean by just by the title. What do we mean by treatment strategies? Well, this is a point where I like to just put a disclaimer and apology. This is the 13th task of Hercules. To actually cover everything that's in the pipeline is just not possible. So what I'm going to aim to do with this presentation is to cover some of the things that I think are going to be important. I'm going to break them up into things that are treatments for symptoms, so symptomatic treatment, but symptoms of physical symptoms, so motor symptoms, and also some non-motor symptoms. So that's the symptomatic side of treatment. And also disease modifying, which I know is a topic that many people with Parkinson's are keen to hear more about. And really what we're talking about there is whether we can get treatments that will slow the progression of the disease, maybe stop the progression of the disease, maybe even reverse some elements of the disease. So I guess, in broad sense, we might be talking about curing Parkinson's disease, which is always a difficult area to get involved with. So that's what I mean by treatment strategies. But what do I mean by pipeline? So I want to sort of break this down again, and really what I want to do is set us up so that for the rest of the presentation you can follow a little bit about my thought processes. So I want to talk about things that are in pipeline across 4 little grades, if you like. So the first grade of that would be, say, well, what treatments are there that have been tested? They've actually been approved, but maybe not widely available. So even in Australia, with the country I practice in, there are many treatments that are available in other parts of the world that haven't yet made it to Australia. So there is a little bit of a bias there. So apologies for those people who might say, well, I have that drug in our country. And a lot of that relates to sort of geopolitics. Has a country got enough money to buy drugs? Is there a universal healthcare system? So those are the things, if you like, that are in the pipeline that are first off the rank. They're actually available now in parts of the world. The second sort of level of the pipeline, if you like, is where a treatment has completed early studies, but further studies are either ongoing or needed, so in the pipeline, if you like. So effectively something that's got a little bit of data to suggest that it might work, but actually it hasn't reached that stage where it would be approved as a therapy. The next thing is an idea where a treatment is being evaluated and we haven't got that first piece of data yet. So this is, if you like, an earlier stage of trial work. So we're really looking to say, okay, well, can we see a signal for a treatment that might be effective in Parkinson's disease? And that can apply to symptomatic and also disease modifying approaches. And then finally, if you like, along this level of pipeline is if a treatment is in development or in fact still on the whiteboard. So the idea is there, but in actual fact we haven't put it into trials just yet. And I will cover some of these elements. But as I say, we're going to go on a bit of a whistle-stop tour. So I'm going to start with symptomatic treatments for physical symptoms of Parkinson's that are not yet widely available, with the caveat some people in the world watching this might be able to get hold of these particular treatments, but not everybody. So I'm going to cover a few of these. There's a drug called safinamide I want to talk about. There's an approach with inhaled levodopa that I want to talk about. There's a non-surgical surgical treatment, focused ultrasound, that I'm going to talk about, and also an advance in our technologies for deep brain stimulation called directional deep brain stimulation, or DBS. So I'm going to go through those next, and the first study that I want to touch upon, or the first drug encapsulated by this study, is a drug called safinamide. So safinamide is a drug that's been licensed, it's being used in various countries around the world, and it's a treatment for people who have known Parkinson's disease, who are running into problems where the tablets aren't controlling their symptoms smoothly throughout the course of a day. And this phenomenon, where we describe this as fluctuation, so people can wear off where they feel their tablets aren't working so well, and they can also get some involuntary dyskinetic movements. So where safinamide comes into the market is it is proposed to have activity a lot like some of the medications we already have available, where it blocks the enzyme that breaks down dopamine in the brain. So this is a monoamine oxidase inhibitor, type B, which is the enzyme that breaks down the dopamine that you naturally produce or you take in your tablets in the brain, extending the period that that dopamine hangs around. So that's the most potent effect, I think, that people have noticed with trials of sulfonamide. And this particular study published in 2017 shows essentially the improvement in those patients who are experiencing periods of off, where their tablets aren't working during the day. And to get into this trial, you had to have significant periods of off, at least an hour and a half, I think, was my recollection of this trial. And effectively what you'll see in this graph is the improvement, or the amount of on time that's been gained by comparing the group who in this trial who had sulfonamide versus those who got the sugar pill. And what you can see from the graphs is that even those patients that received placebo did improve the amount of good on time that they had during the day, but there was a significantly greater improvement in the group with sulfonamide. Now, this is a trial that didn't compare its effect to other drugs, so we don't know whether adding combination of other medications would have had similar effects, but this is common practice for clinical trials. So I think we can be confident that the sulfonamide is a drug that can, if you like, complement patients who are struggling with these off periods where they're probably taking levodopa and they will need something to, if you like, extend the life of that medication. Not terribly different to some of the other monoamine oxidase inhibitors that we have, such as selegiline, otherwise known as eldepril, razagiline, otherwise known as azilate, but a useful addition. One of the other elements of this drug, sulfonamide, that people have talked about is its anti-dyskinetic properties. That is to say, can it slow down those involuntary movements? And the bottom line is that's a different pathway, and it's said to block the glutamatergic, so one of these other pathways that goes wrong in the brain in Parkinson's disease. Now, the trial data that exists today does not confirm that this drug is very potent at reducing those dyskinetic movements, but I think a number of people are suggesting that there are more trials needed. Okay, so moving on from sulfonamide, one of the other treatments that has been licensed in parts of the world is the idea of an inhaled levodopa preparation. So most people will know that levodopa is the cornerstone of therapy in Parkinson's disease, whereby you take your drug and that level goes up, replacing dopamine in the brain. But of course, in those wearing off periods that we were just talking about, we don't have something that acts very quickly. Simply and quickly, I should say. So at the moment, we have strategies where we might say, well, let's try a dispersible form of levodopa, mix it in water, take it. Perhaps it acts a little bit more quickly. The data suggests it's still going to take at least half an hour before it gets from your mouth down to your stomach and then back to your brain. And of course, the other effective therapy, which would be subcutaneous apomorphine. So an injection of a dopamine agonist, which within certainly 20 minutes, and probably for most patients within the first 5 to 10, will switch them back on from their off state. But of course that involves an injection. So this advance, which is covered in this particular trial, published only in 2019, shows that actually there may be a way of improving this, if you like, turning on pattern in patients who are switching off using inhaled levodopa. So not having to inject, but maybe to actuate, a bit like you do for asthma and other lung conditions. So in this trial, what you can see presented here are the main effects of the study. And in this study, what they did was have a placebo group who inhaled something that wasn't an active drug, and 2 doses of the active drug. And what you'll see in the panel on the left here with the graph, the bar chart, is that in patients who are on placebo, much fewer of them, when they inhaled, actually switched back on. So not zero though, interestingly. So 36% of patients, when they took the placebo, seemed to switch back on. Now, whether that's because the dopamine system kicks in as part of the placebo effect, which we do understand a little bit about, but as you'll see from the other 2 bars on the chart, you'll see that there were significantly more people who were taking the active drug who actually switched back on compared to placebo. And that was coupled with an improvement in the physical rating scores of those patients taking the inhalers. And again, this is now a different type of line, kind of a graph, where you can see that the placebo doesn't seem to get as better, and the improvement in physical symptoms is noted by, if you like, a lowering of the score the physical unified Parkinson's disease rating scale. And you can see that the two active arms of the drug, different strengths of the drug, both improve those physical ratings. So this is a therapy, again, a strategy that's available in some parts of the world, not yet universally available, and certainly there are other strategies that are coming. People are looking at intranasal delivery of levodopa, and so this may just be the first one of these approaches that hopefully will get more. So the next trial that I want to talk about is in relation to physical improvements, is this non-surgical surgery. And people may have seen my other videos on focused ultrasound talking about where it's used in essential tremor, and I hinted at that point that there were trials ongoing for its use in Parkinson's disease. And here we see just one of those published in 2017, where essentially patients with Parkinson's disease, but particularly tremor-dominant Parkinson's disease, underwent this procedure. So in this procedure, what happens is that the doctors take some very detailed scans of the brain, and then over the top of the head, without any need for surgery, is a device which can focus ultrasound onto specific regions of the brain, burning those cells and, if you like, disrupting those circuits that might be causing tremor. And here you can see on the photograph the sort of setup that we see for this focused ultrasound. And on the left-hand side, you can see how this therapy was used. It's quite a small number of patients. I think about 27, if I remember rightly, were involved in this trial. But you can see, compared to having a sham procedure where you went through all of this but didn't have holes burnt in the brain, you can see that there were significant reductions in the amount of tremor that these patients had when they actually had the lesion, this burning of the hole, in the brain. So this is a very positive approach, no surgery, but there are some caveats. The first, of course, being this is not a universal treatment for Parkinson's disease. It is useful only for those patients who are struggling with tremor that does not respond to other therapies. And it could be argued, perhaps, that if you were fit enough to have deep brain stimulation surgery, you might be better off having that approach, because unlike where you take a hole in the brain, the DBS, the deep brain stimulation, may actually be adjustable. And that might also deal with some of the other non-tremor features that we get in Parkinson's. But certainly this is a very useful approach, and the effects were sustained over time. Interestingly, again, even the sham procedure, those patients who didn't have holes, you see an improvement in their tremor despite having not had the non-surgical surgery. So again, some important information for us about the way we need to set up trials. We mentioned deep brain stimulation. It's a therapy that we've had now for over 25 years, where electrodes are placed in the brain and they emit an electrical current, stunning those parts of the brain where the current flows through it. And you'll see in this panel the conventional electrode, where electricity, if you like, spreads like a sphere. And that's good for treating some symptoms, but may actually cause side effects if you didn't want various parts of the brain stimulated. So this new approach of directional DBS, where you can have electrodes that, if you like, can funnel the electrical field into a particular direction, the hope is that this will lead to more dedicated symptom relief, with less in the way of side effects. So these therapies are available in certain parts of the world and are becoming more widespread. I wanted to touch upon non-motor symptoms, and really, I guess one of the things that's most close to my heart is the understanding of psychosis, hallucinations, delusions. And this is a trial that was published now nearly 5 years ago, in fact over 5 years ago, where patients with psychosis as part of their Parkinson's disease were treated with a novel drug treatment called pimavanserin, which works in a different way to most of the drugs that we use for this particular complaint. It works through a different neurotransmitter system, but I really wanted to just highlight that it does appear that in this sort of short trial that this drug can have a positive benefit to those patients who are having psychosis as part of their disease. Again, unfortunately, this drug has not made it across most of the countries in the world, but it'll be very interesting to see whether these sorts of approaches could come and improve the quality of life for people we know who are struggling with these symptoms, usually in the advanced stages of disease. Okay. What about symptomatic trials where symptomatic therapies where we need more treatment? So I'm going to talk now about a number of other trials. We're going to talk about Camicanal, Neuroderm, or ND016 I think it is, pluripotent stem cells, and also glial-derived neurotrophic growth factor, GDNF. So these are all a little bit different, and I'm going to take you through them one by one. So the first is a drug called Camicanal, and Camicanal has been used in a small trial, I think again about, I don't know, 30-40 patients maybe, and this drug acts to improve the emptying of the stomach. So we know as Parkinson's advances, the stomach gets slower, sluggish, and you get what we call gastroparesis, which means that tablets may not leave the stomach even though you take them on time. The acid gets to them and they're no longer effective, and we get pill failure. Camicanal is an agonist of motilin. Motilin is a hormone that tells your stomach to empty and get things moving along. So this is, if you like, pretends to be motilin. And the idea is that it might therefore promote gastric emptying, so stomach emptying. And this phase 2 trial, which had 38 patients on Camicanal and 20 patients on placebo, went for about 7 to 9 days. And what they actually did report was that patients who had the active treatment had improved periods where their tablets seemed to be working, less periods where they were switched off, and their physical symptoms seemed to have been improved. But the authors of this study acknowledge it's a very small piece of work and there are a lot of other factors at play, so I think the idea of taking this into a bigger trial is now something that's very actively being pursued. The Neuroderm, or ND0612, and there are lots of little letters on the end, is a different approach altogether, again in earlier stages of the trial work. And you'll see here some of the work that's been published in abstract form, suggesting that it hasn't yet been through the rigorous scientific process. But the idea here is that maybe we could get a more constant or consistent delivery of levodopa therapy if it was administered under the skin via an injection, a little bit like apomorphine is. This would of course bypass that problem of gastric emptying, so the slow stomach, the gastroparesis, and effectively deliver the drug directly, which would be absorbed and hopefully go to the brain. There are ongoing research works going ahead with looking at these trials, and as you can see here, these are pictures lifted from the company who are producing this approach, just to give an idea, a concept of what they envisage. So this is something again that we're waiting to hear more data upon. I wanted to briefly talk about pluripotent stem cells, and I will plug my own work, and this is a fantastic interview that Ryosuke Takahashi from Kyoto University gave me when I was in Japan just last year. Ryosuke is the clinical lead for this landmark study where people are using induced pluripotent stem cells. So let's just go back a stage. So Parkinson's disease cells are dying. The population of cells that probably cause the most of the symptoms, certainly at the start of the disease, produce a chemical called dopamine. So the idea of a stem cell would be to take a cell and make it produce dopamine. And in the old days, what we did was a number of negative trials where people harvested cells from unborn babies, fetuses, that were going on to produce dopamine and put them into the brains of patients with advanced Parkinson's. Now, all of those trials were negative. They were deemed to be dangerous because patients developed involuntary movements that needed surgery to correct. The idea of an induced pluripotent stem cell is to try and take cells from the person themselves, the patient themselves, process them in a way that means that they'll be, if you like, a better fit for the individual patient. Now this work has started. This photograph, as you can see, was the front page of the Japan Times. It made the front cover there. This was the first patient who got operated on just the day before I arrived in Japan. And if you want to go to the link where I interview Professor Takahashi about this, you'll see the link on the slide, and I encourage you to do it, especially if you're interested in stem cells. But what you will find Professor Takahashi saying very clearly, stem cells are not a cure for Parkinson's. They are there to essentially replace tablets. They're a way of getting dopamine into your brain in hopefully a more physiological fashion. That is to say, the brain, essentially having a more constant level of dopamine in it. The other thing that he warned people about was that just to do, I think, 7 patients is going to take the best part of the next 5 years. So it will be a long time before we get to learn about how effective this therapy is. The study is restricted to patients in Japan, and the bottom line is that even if that trial is overwhelmingly successful, the idea of scaling that process up, the reason they can only do that number in such a long time is that the science behind producing these particular cells is very complicated, and to scale it up is going to be a mammoth challenge, but hopefully something to keep an eye on for the future. I also wanted to talk about these glial cell line derived neurotrophic factors because it's a topical story that just came out in 2019 this year, where effectively researchers in Bristol University who've actually been using this approach for some time with some negative studies in the past have gone back to revisit this. So if we take the example again of the cells dying in the brain, the idea of a growth factor is it may be something that we could add to the brain to re-stimulate growth of those cells so that they could actually be healthy again. So the growth factor that's being used, this GDNF, was trialled previously in Parkinson's, and the idea was to sort of deliver it deep into the brain of advanced Parkinson sufferers. And that trial was negative, but it was felt that perhaps the problem was that there wasn't a consistent delivery of the growth factor. And in this trial, what people have done is to try and take a small number of advanced patients, about 35 in total, randomized to either getting the growth factor or not getting the growth factor, but to deliver the growth factor in a unique way over the course of 40 weeks. So not a long trial, about 9 months in total. So maybe too short to get a positive result, but people, very keen to know about this trial. And this is really the headline story. So essentially those people who were given the growth factor compared to those people who were given the placebo, there were no differences in their outcomes over the course of the 9-month trial. What you'll see on the left panel here is some of the setup, how the science was done, and you can see the idea of putting in a little plug, a cannula, where effectively the drug could be delivered, and I think patients had monthly delivery of dose into the deep parts of the brain, trying to encourage this these dopamine cells to regrow and to function. So a very interesting approach, something that obviously a negative study again, but something that I think that people will want to pursue further. Okay, we're going to move for the rest of the talk across to the idea of disease modification, and we're really talking about can we slow, stop, reverse the disease, probably not reverse so much in this section. And this slide, taken from work published by one of my former colleagues, Tom Faltony, and his Dr. Atuda, essentially covers what we know might be potential targets for us to look at in terms of modifying Parkinson's disease across a range of different strategies, and I will cover as many of these as I can, as coherently as I can. I'm going to start from a basic starting point of genetics in Parkinson's disease. So what are the genes telling us? So it used to be very exciting to sort of think, well, do I have a gene that causes my disease? And although that's helpful, it doesn't really take us much further forward in terms of what is it the gene telling us about the disease, not just I have my disease because of this gene. Genes encode proteins, and proteins are the building blocks of stuff inside cells at the cellular level, at the microscopic level. They can build the wall of various structures. They can be enzymes that do things, make things go faster, break things down, build things up. And the idea behind a genetic abnormality is that sometimes you might have a gene mutation that causes an overexpression. So you might have too much of a protein, might be getting in the way, gets tangled up in the safest way to deal with too much protein. And we see that with a protein called alpha-synuclein. I'm going to talk about that a little bit later on. And there's also underexpression, so you might not have enough of the correct enzyme to clear garbage from cells. So this may also impact on other regulatory pathways, whether it's energy pathways in the brain or inflammatory pathways. If you don't have enough of that enzyme because you have a genetic mutation, then that may be a problem. One of the enzymes that I'm going to talk about, which is related to a genetic mutation, is glucose cerebrosidase, otherwise known as GBA. So GBA clears garbage from the cells via a structure called the lysosome. If you like, it's the recycling depot and gets rid of the garbage. If you have a genetic mutation where you haven't got one of these genes from mum or dad that works properly, you'll end up with a rare storage disease, storage because proteins accumulate inside cells, called Gaucher's disease or Gaucher's disease, depending on how posh you are or how posh you are or where you were trained. This is a condition that is very serious, usually causes death in childhood. There are some adults with this, and in fact they gave us a little bit of a clue in the Parkinson's world. And as often with genetic mutations, you can see higher proportions of people where there is often, if you like, a closed community. So the Ashkenazi Jewish population would be a good example of where we see Gaucher's disease. But what we found from studying people with Gaucher's was actually, it looked as though if you had just one bad copy, so not both mum and dad, but one bad copy of the gene, that was associated with people who had Parkinson's. About 5 to 10% of people who have Parkinson's disease will have one bad copy of this GBA gene. So not enough to give them Gaucher's disease, but possibly, it doesn't necessarily mean that it causes Parkinson's, but certainly a clue as to where we should be looking. And this is interesting work published by my own team, Nick Zamko, who leads this at the University of Sydney, looking at, okay, if you've got just one bad copy of the gene, you'd expect to have a problem with enough enzyme. So what we did was a trial where we looked at patients who didn't have any problem with that gene at all. And what we found compared to people who were healthy was that actual fact Parkinson's disease patients who didn't have any genetic mutation still had lower activity of the enzyme that gene would produce, this GBA, glucocerebrosidase enzyme, suggesting that in not just the Parkinson's patient with a genetic mutation, but in all Parkinson's disease patients, enzymes or the pathways that the genetics uncover may be an important clue as to what to do in terms of treatment. And this has been taken into this graph, and this is the lysosome. I'll just show it again, hiding there in the middle of the cell, clearing garbage. So the idea that's been taken forward in clinical trials, and I've put a number of these clinicaltrials.gov, and this is the website link you'll see across the trials I'm going to cover. Essentially, this is one of them called Moves PD. So this is an enzyme approach by a company, I think Sanofi is the company, assessing a drug called venglustat. And venglustat essentially blocks the enzyme that produces glucocerebrosidase. So the idea behind this trial is to take patients who have a known genetic mutation, so they've got a GBA mutation, and give them this agent which will block the production, if you like, of the rogue chemical that's building up that needs the enzyme to clear it. So this is an approach that's limited only to those patients with the genetic mutation, GBA mutation, and there are only 200 patients, and as far as I am aware, we've tried to get the studies across in Australia, but they're running in other parts of the world at the moment. This study is, I think, due to report in the next year or so. Often these times can blow out, but watch this space. The other approach looking at the action of the glucocerebrosidase is this AIM-PD being led by Tony Shapiro and his team in London, and also a team of researchers in Canada who are also based in London. It's known as the Two Londons study. And this is an interesting approach because it's the first thing we're going to talk about with repurposing. So ambroxol is part of cough linctus, a cough medicine that's out there, a chemical that's in that, which we know can actually increase the levels of the glucocerebrosidase enzyme. So this is interesting. We're tapping into, if you like, that pathway by repurposing a drug that was designed for something completely different. And this trial is not so much looking to see the efficacy, but to see, if you like, the biology, the impact. So they're going to compare 10 patients who've got the genetic mutation with 10 who don't, who are matched on other clinical variables, to see whether administering ambroxol can impact some degree the symptoms, but more importantly the biological processes going on. So this is a trial, I think they've closed their recruitment at the moment, and we're yet to hear the report of it again, should be imminent. So I want to move from this particular enzyme back to something I touched on earlier, which is the idea of Lewy bodies. So Lewy bodies essentially are tangles of alpha-synuclein protein, one of the proteins I mentioned in connection with genetic abnormalities. So people with Parkinson's disease will have been told at some point they might have Lewy bodies in their brain, and this is essentially what people look at down the microscope and see as tangles of this alpha-synuclein protein. And it's found inside those dying, predominantly dopamine-producing cells. So we don't know if it's the villain or the hero, but effectively it serves as something that we can target, because again in this schematic we can see here there are ways where this tangles of alpha-synuclein may accumulate in cells, it may spread from cell to cell, and so it allows us some opportunity to try and treat the disease. And it comes into this territory of what we call a Parkinson's vaccine. So vaccination, people might be familiar with the idea of active vaccination and vaccination programs, where in this instance what we might do is take a little bit of abnormal alpha-synuclein and inject it into, I don't know, children who don't have Parkinson's, all of the children, like we do with other vaccination programs. And the idea then was that the child or individual might see that fragment of alpha-synuclein, produce their own immunity to it, so that in later life they may be able to rebuff a Parkinson type process. So this would be easy to upscale if we get the right thing working and provide herd immunity. And there are models of these vaccine approaches that have been generated in mice, but I think mice are a long way away from humans. As people will know, humans are the only animal on the planet that get Parkinson's disease. But what about what's happening right now in terms of clinical trials? And this is the idea of targeted immunotherapy. So passive immunization, as opposed to generating an immune response, we actually can give people directly the antibodies that we think they might need. So effectively, we have these approaches for things like rheumatoid arthritis, inflammatory bowel disease, and the idea would be to say, okay, well, you've got Parkinson's, we just diagnosed it, or maybe even before we can diagnose it, we would then give you the treatment. Now, how expensive this is, how much treatment we give people, do we give it once, do we keep giving it, how often do we give it, are all questions that are yet to be answered. But there are lots of ways we could think about it. Do we break up the tangles that are there? Do we try and just stop them from aggregating in the first place? Do we stop them from being transmitted from cell to cell? So there are lots of different targets that we could go for. And the first data that's come out of this approach, where we use, if you like, a targeted antibody approach, what we call monoclonal antibodies, deliberately targeting the synuclein protein, was just some safety data released in 2018, where effectively a small group of Parkinson's patients were given these monoclonal antibodies, and they reported at 24 weeks the safety. And they reported that it seemed safe, they didn't seem to have any adverse reactions, and they have recommended a proper constructed phase 2. And there are a number of these monoclonal antibody approaches currently being launched, generally by large pharmaceutical companies who have the resources to afford to do these trials. So what about the other approaches? Rather than targeting alpha-synuclein, what about the other, if you like, effects that are going on? There are lots of things that are going wrong inside the cells in Parkinson's. Oxidative stress, how much energy the cell has, governed by how well the mitochondria, the little power packs inside cells, are working. The regulation of the membrane, we're using things like calcium to make sure that the electricity across the divide is normal, but these things are active, they need energy, these can go wrong. Inflammatory pathways can go wrong. Programmed cell death, so cells are supposed to die, but perhaps it's too much. And this can happen through a number of processes, including iron-related processes called ferroptosis, so apoptosis, ferroptosis. So lots of different approaches here sort of contained in that part of the schema of the diagram. And what I'm not able to do is to dive into the details of what's going on in all of them, but just to touch on some of the trials that are out there. So the first trial I want to talk about is exenatide. So exenatide is a subcutaneous treatment that's used for diabetes. So again, a repurposing drug strategy. And it's thought that perhaps part of what goes wrong in Parkinson's is some signaling within the brain's insulin pathways. Within the pathways inside each individual cell, there's this AKT activity, a critical protein that's involved in cell survival processes, and this exenatide seems to enhance the activity of that. And it may also be working through neuroinflammatory pathways. Most of this has not been well worked out because, of course, doing studies in humans is very tricky because you need to take the cells out, and these are living patients. So a lot of this is based on animal work. However, there is a phase 2 trial. It was a small trial with just 60 patients conducted at one centre in London, and this is the main result from that trial, published now a couple of years ago. Again, led by Tom Falsey, my old colleague from Cambridge, now working in London. And what you'll see on this set of graphs, and probably easier just to focus on the panel on the right, so B, is that if you were injecting yourself with a placebo, then you continue to steadily just get worse. That's that red line. If you were injecting yourself with exenatide, however, there seemed to be an improvement in your symptoms, then it seemed to stabilize. And when they took the exenatide injections away, things seemed to get worse, but you weren't as bad as if you had had just the placebo. And this obviously is exciting and has spawned the idea of another bigger trial. Obviously, people might say, well, that drug's available now, Simon, what should we do? And I thought I'd prevent that question, or preclude that question, by asking Tom Falsey himself. So this is us at a conference after he knew the results. And basically what he is saying is he is not giving this treatment to any patients. It's a very small study, needs further data. There is a large group now working on this in collaboration with the Link Clinical Trials Initiative. Cure Parkinson's UK are involved, and my understanding is the next trial will be a bigger study across multiple centres, probably running for a longer period, like 2 years. So we wait to see. We're very excited. Other approaches, ursodeoxycholic acid. These are all about improving neuronal functional survival. Again, I put the reference there so you can look up these trials. They're in the public domain. And ursodeoxycholic acid, UDCA, is a treatment that's used for primary biliary cirrhosis, a condition of the liver, but it's thought that it might enhance mitochondrial, those power packs of the cell. And there is a phase 2 trial going on, I think in one centre at Sheffield. There are only 30 patients, and there are only patients in the first 3 years of the condition. So this trial has yet to report. But somebody approaching this idea again, repurposing. Inosine, essentially one of the things that people need to know about inosine is that in Parkinson's disease, what we have discovered is that if you've got higher levels of urate related to uric acid, which is what gives you gout, you seem to have less chance of getting Parkinson's, or if you have Parkinson's, it seems to progress more slowly. Now, why that should work, we don't know. We think that this may be through an antioxidant response pathway. And what's interesting is that inosine, or inosine, is the precursor of urate. And so you can actually take inosine as a supplement, boost the level of urate, and hopefully impact upon the disease. As you'll see, there is a clinical trial going on here. It's actually gone up to phase 3, so larger numbers of patients involved, 270 patients across centres, I think mainly in North America. And again, recruiting patients in the earlier stages of their disease, less than 3 years. So this trial, again, hopefully will report in the next year or so. Isradipine. So isradipine is a blood pressure treatment, and what we've seen in the past with patients on blood pressure treatments, or certainly individual types of blood pressure, these dihydropyridines, is again there may be lower rates of developing Parkinson's disease. This drug acts on that calcium homeostasis that we were talking about, regulating what goes across the cell membrane, improves mitochondrial function, and there is currently a phase 3, again 336 patients, less into their disease, less than 3 years of disease, again going across centres in North America, another repurposing approach, yet to report. And finally, not finally, but nilotinib, a treatment for chronic myeloid leukemia, works through this apoptotic pathway where cells are programmed to die. There is a small phase 2 trial going on across centres in North America. I think that's again going to focus on patients in the first 3 years of disease, probably not going to report for the next year or two at least. A drug that needs to be carefully looked at because, of course, it can have adverse side effects, including very significant cardiac or heart problems. So the safety is going to be very important with these trials. The other approach I wanted to talk about was deferoprone. We mentioned ferroptosis, so this programmed cell death relying on iron. And deferoprone is a treatment that's been available for patients with thalassemia now for several years. And it believe that it might modulate ferroptosis, and there are studies going on, including here in Australia, looking at whether deferoprone may be useful for slowing the course of disease, again targeting those patients who are in the first 3 years or so of their disease. What about on the whiteboard, things that haven't yet made it to clinical trials? So maybe you'll hear more about this elsewhere, but I want to talk about antisense therapy. It's a wonderful term, isn't it? Antisense. But what we're talking about here is things that might be useful in treating diseases that are related to genes. And we've seen this approach work in other neurological diseases where treatments are now available for rare conditions like spinal muscular atrophy, a very proud Australian group leading that charge. And effectively, the idea may be applicable to certain patients who've got genetic mutations in Parkinson's. So what we know is that those genetic mutations will produce something that signals that you're going to make, I don't know, a protein that becomes an enzyme. It doesn't really matter. But the idea is that we know what the signal that protein or that gene must be encoding for to get to that protein. So what we can do is to make an antisense molecule, whether it's a little molecule of DNA or RNA, things that, if you like, encode what's going on in your genetic makeup, or indeed a chemical analog that goes and hunts specifically for that signaling molecule, holds onto it and stops it from producing the mutated form of the enzyme that you don't really want. So we'd ideally, in that situation, maybe turn off the mutated gene. We could, of course, turn it on if we thought it was underactive. So there are ways that we could maybe look at these approaches, but still on the whiteboard. The other thing that I want to just touch on that's still on the whiteboard are nanoparticles. I love the idea of talking about nanoparticles. So these are incredibly small molecules. And this is, if you like, a magic bullet approach. And people will be aware of some of the nanotechnology where you can make a tiny little motor, it'll drive around the bloodstream, find cancer cells. So these are things that people talk about. They're hopefully science future rather than science fiction, but at the moment what we're talking about is the idea of these graphene quantum dots, which are so small that they're beyond the resolution of most microscopes. But the idea is to say, well, look, if we could get one of these graphene quantum dots and target it towards alpha-synuclein as our main bad guy, perhaps we could stop these fibrils, these strands of bad alpha-synuclein clumping together, and even get them to disaggregate, stop them from tangling. So this is on the whiteboard. It's one of those things that I flag because I think it's interesting that you know that people are trying to find new approaches. So what about the path to a cure? So we've had advances over the last 15, 20 years where we've got more genetics, more epidemiology, more basic science. So we have more of these cellular pathways, and we've also got people working together more. I mentioned briefly the Linked Clinical Trials Initiative, dragging in people like the Shake It Up Australia, Parkinson's Australia are represented, Cure Parkinson's UK, Michael J. Fox, Van Andel, Garvan Institute, and others, including the University of Sydney. Essentially trying to come together to design better trials, trial the right agents, cooperate more, which is what patients really want us to do. What about Australia's role? So we know that we've got lots of patients who haven't been able to get into trials. As I mentioned, a whole bunch of them are elsewhere in the world. We've got lots of partners, national and international, and we've got doctors, nurses, allied health people who are clinically very well trained, very keen to get involved in trial work, and we've got some of the leading scientists in Australia in the field of Parkinson's disease, many of whom you can bet your bottom dollar are going to be making major breakthroughs in this area. So I'll just touch briefly on the Australian Parkinson's Mission, which essentially is a federally funded program of disease-modifying trials. So the federal government of Australia in 2019 put forward $30 million over 5 years to allow Australia to get into this space. So the plan will be to have a program of trials essentially targeting disease modification. And the idea will be to try and screen as many drugs as we can, as quickly as we can, to look for a signal that might be useful to take to larger trials, where it probably will go international, multi-center, all of those things, but also to combine our clinical trial work with clever science. So the bottom line is that we know that there are probably some genetic influences. We know that we have some markers for whether the target we think is being engaged. So if it's an anti-inflammatory approach, maybe we can look at inflammatory markers. If it's an approach that looks at energy stores, we can look at those things. But we can also look, because of some of the clever scientists here in Australia, at whether we're engaging the disease. I showed you those activity, the enzyme activity in patients with Parkinson's disease, and whether we can improve those markers at the time of screening a drug would allow us then to have more confidence and say that drug should definitely be in a trial, whereas this one that didn't seem to engage with the disease, maybe not so important. And the idea will be to try and have lots of different drugs in the same trials to start against less people taking placebo. So effectively the idea will be to have lots of drugs being trialled and hopefully the minimum number of patients on a placebo. It will be a program of trials. Essentially, we need to learn from each of them. We need to have them overlapping. We need more centres across Australia to do this. And we have got people in metropolitan and in regional sites who are keen to get involved. We know that patients are mustard keen to get involved. The idea, as I say, will be to sort of have this approach where multiple arms are running, patients are being assessed at variable points throughout the trial, and we'll be doing various assessments, but nothing too complicated. Maybe taking medications away before you come into the clinic, having blood tests allowing you to have your medications in the clinic, but effectively something that will allow us to screen these drugs quite quickly. So we would have different drugs, different types. We don't want to have all anti-inflammatory or all something that's in a target energy. So different types of drugs or different types of approaches. There'll be no brain scans, no need to do spinal fluid. Blood we will take for our biologicals. And if you are interested in getting involved in these trials, living in Australia, then the website is www.theapm.org.au. So in summary, we've got this problem. Parkinson's is a growing socioeconomic challenge. We have some solutions, potentially. We've got lots of very willing patients and clinicians and scientists. The idea of trying to rapidly identify cures or better treatments with combination of precision medicine. So the idea really from here is to go forward and try and get as many people involved in trials as we can, so that we can get past our current impasse of having no effective medication for Parkinson's disease. And with that, I'm going to close and put up a holding slide which has got some resources on there so that people can look at these videos, register for the websites, and if needs be, reach out and follow me on Twitter. Thank you so much for your attention.